can all hear me well? Yes. Great. So when they asked me to do science and cocktails, I was worried a little bit because I figured after 15 minutes, everybody would go like, you know what, I want to have cocktails now, so hurry up. I realize now that you've already started drinking, so this is great. That way, I'm not under any type of pressure, and on top of that, you're going to be less critical, right? So what I'm going to talk about to you today is, well, the title is, I can read it from here, How Evolution Creates Complexity from Viruses to Brains. Now, from viruses to brains means pretty much anything, right? Indeed, I'm going to talk to you about trying to understand all of life on Earth and suggest that, well, maybe if we really understand it well enough here, can we say something about life away from Earth? What you see actually here is the cover of my book, and its main title is actually The Evolution of Biological Information. There are two very important concepts that are in that title. It's the word evolution, and it's the word information. These are two fundamental concepts. One is a 19th century concept, namely evolution, and one is a 20th century concept, information. And what I'm going to tell you about is how you can meld these two concepts together to get a 21st century understanding of essentially the biosphere that we're living in. So let's take a look. What is it that we actually are trying to understand? And it's this. You see here, two mammals within, a forest. And of course, that looks very familiar to you. It's not something where you go like, wow, you're familiar to it. But think about any other planet that you've ever seen the surface of. Think of the moon or the Mars. They don't look like that at all. I mean, at all. So there's something very different going on that gives you this, and then gives you this, and this, and this, and this, and this, and this. This is not what you would normally find, and you will not be able to use 18th century and 19th century concepts from physics, from chemistry, and from astronomy to make you understand this. Now, I've shown these pictures for you in an order. In fact, the one that you see right here, you might remember that from 5 years ago. This is the COVID virus, okay? These are bacteria, and you see, in a sense, I went backwards in complexity, however you want to define it. This here is a trypanosome. It's a unicellular eukaryote. Here is a very simple multicellular organism called Volvox, a jellyfish, which is an invertebrate. Here's a butterfly, and there we are. And so there's this concept that somehow there are different levels of complexity. Nobody would think that hit the wrong button here. Nobody would think that the level of complexity of mammals, including us humans, and the virus are somehow the same. So can we understand, first of all, how all of this emerges? And what is the relationship between them? Is really one more complex than another? Can we actually attach a number and have a ranking between them? But to understand how the relationship between those, we have to come up with some concepts that are very different from those of physics, mathematics, and astronomy. So I want to first talk a little bit about how the concept of evolution even emerged as a new idea, the new idea that changes everything. And so I'm going to have to tell you a little bit about history. When we are considering the times when people were really not sure about how any of this works, there was, in fact, a theory that told you that it was all solved, that there was nothing to understand. Namely, that all of this was created at one time, well, during one day, so to speak, 6,000 years ago, created by God. All of this at the same time. And that there are no questions to be asked. Because there was a fundamental church doctrine that told you about how old the universe is, how old Earth is, and how all forms of life was formed. And if you were curious about this, you're not even allowed to think about this, because there are, strong penalties. Galileo was obviously very, aware of that, and there were certainly strong penalties for people who would question that theory. So the intellectual guiding line was really this one. It was called the Great Chain of Being. This here is actually from the 16th century, and it's essentially showing the ordering in complexity of all life on Earth, all created, of course, by the divine creator. You can't see exactly what's on there. I'll show you. So down there, for example, is the devil. Okay? Above it are like rocks and minerals. You see that. But in this row here, we have plants. And above that, there's fish. And then we go higher in the great chain of being, according to this. And we see some birds. Then there's people. Then there's angels. And above it, of course, is God. Now, it's a not very consistent ladder, because I left out something there, a row, which is weirdly between plants and fish, and it's got unicorns on it. And so from a scientific point of view, you don't get an A for this, right? However, it was what you were supposed to believe. Here's a poem from Alexander Pope from the 18th century, Essay on Man, where he writes about the great chain of being. And he says, vast chain of being, which from God began, nature ethereal, human, angel, man, beast, bird, fish, insect, what no eye can see, no glass can reach, from infinite to z. So with what no eye can see, of course, they mean microbial, things like, and no glass can reach means no telescope. So it's like on all scales, but it's all from infinite to to God. That is the atmosphere that everybody was in at the time. And it created an intellectual prison. Because, who wants to actually risk communicating about their ideas when, that was something that could risk your life? So in order to understand how a revolutionary idea like evolution can even emerge, we need to understand that it cannot emerge at all once, fully formed. In fact, we don't even know from a fundamental point of view how new ideas and theories are formed to begin with. And the reason for that is that there is no logical way to do that. Logic operates from deduction. You can deduce facts from a theory, but you cannot, by any logical process, create a theory from facts. So we don't even know how that works, but one thing we do know is that you have to prepare the ground for that, so that the steps are small enough, right? And so I'm going to give you 6 vignettes of people who've actually prepared the grounds to see, in a sense, how many different steps were needed to finally get to the point where can create the big leap. So these are the following. So we're starting actually in the 17th century, 17th to 18th century, and I'm showing you a book written by Benoît de Maillet, who is a French nobleman. You've probably never heard his name, and he's not famous now. But he was very famous when this book was published in 1750. And you will notice that it was published 12 years after his death. Why? Because it contained some things that would have gotten him killed. In fact, he wrote it as Teliamet aux entretiens d'un philosophe indien avec un missionnaire François. It means dialogues between an Indian philosopher and a French missionary. He, of course, would, as a dialogue, play the role of the French missionary who is fearing. Teliamet is the Indian philosopher who says things that you can't possibly say about the formation of the earth, about where men came from. He realized that on top of mountains there are actually sediments that are clearly seashells. And then how could the seashells get to the top of the mountain? Well, that means that the mountains either form or there in fact was a flood that retreated, la diminution de la mer. But at the very beginning, it's clear that the author is absolutely pissed at the church. It talks about how they dare to, put you into this stupid prison. The word stupid literally appears in the introduction. It would be, it's fantastic reading now when you're seeing like an anger lashed out like that by saying, it's like, how can you believe things like that? But however, he could not publish it in his lifetime. And many of the things he wrote about were fantastical about mermaids and mermen. But the important thing is, he actually looked at some evidence and he started thinking and he started questioning. And so that started sort of a loosening of these the bars that were the intellectual prison. Let's go 100 years later. This is Carl Linnaeus. You've probably heard of him, and particularly here in the Nordic countries. He wrote a book called Systema Naturae, where he, in fact, has created the system of classifying organisms and plants and things like that by the genus name and the species name. And that is a very important step, because if you don't even know how to count, how to see differences between things, how can you talk about diversity and where it comes from? So you first need to establish a way to count things and to measure differences. The other advantage that I was giving is that rich people realized that they could have their name attached to a new species if they funded expeditions where they find them. So suddenly there were all these expeditions because people, wanted to have their names attached to it. So it creates the wealth of knowledge that you otherwise would not have had. So at the same time, we have here the Comte de Buffon, Georges Leclerc, and while he didn't do classification, he described everything that he could get his hands on. He wrote 60 volumes of descriptions of animals, basically from the simplest one you could find all the way to the most complex ones. And with beautiful pictures. However, he didn't want to have just pictures, so he did also a lot of writing. And it's almost like when you read there that at some point he gets bored just describing things, and he starts thinking about, well, where could all this stuff come from? And if you read closely, you'll find that the central ideas of Darwinian evolution are already contained in some of the pages. Except he never really made a theory out of it. He never really understood the fundamental importance. So he didn't say, well, once we understand that, what can we learn from it? It was almost like, like I'm bored, let's talk a little bit about some other things. But it was hugely important because the times were different then. These books were essentially in the pocket of any educated person. Because keep in mind, there were no cell phones, there was no Netflix. People were reading books when they were educated enough to read. I actually have 25 volumes of Buffon in a format that is smaller than a cell phone, because people would just keep it in their pockets and started reading it wherever they were. So the Buffons, this stuff was super famous. He was super famous, even though, he just cataloged stuff. So let's move a little bit further. Here is Jean Lamarck. You are probably familiar with his theory of evolution, the Lamarckian evolution, the one that's famously wrong, because he was theorizing that the change in characters comes from the use and disuse of a character. However, even though that part of the theory is wrong, many of other parts of the theory were fairly similar to what Darwin later would come up with. The important thing here, however, is that he literally was one of the first to say, I'm going to present a theory of where this stuff comes from. Never mind it was wrong. It's an important step because somebody needs to take these steps and then, well, if it was wrong, let's elaborate on it. But it was an important step. He was a contemporary of the next fellow, and to some extent was eclipsed by this guy. This guy was way more famous. They worked side by side at the museum in Paris. But Cuvier was a showman. He had understood that if you're looking at the skeleton of an animal, for example, then because all of the parts of the animal are so adapted to each other, that if you know one of them, you can predict all the rest of it. That, in a sense, requires you to understand that these things are a whole. And so he would make a show that somebody would give him a bone and says, tell us about the rest of the animal, and he would do that. And so he got famous for that, and literally started this concept of paleontological reconstruction of skeletons. So again, very influential, and in particular, he showed incontrovertibly that some of the bones come from animals that do not exist anymore, that are extinct. That is absolutely contrary to the accepted worldview, which said all species were created at the same time, in the same form, nothing new comes about, nothing goes extinct. He incontrovertibly showed that's nonsense. It's like a dagger in the heart of the accepted church doctrine. And so that's why he's such an important figure in preparing the ground that came next. But before I get to Darwin, here's another important figure, that's Charles Lyell. He's a geologist, or was. He wrote the book called The Principles of Geology in 3 volumes. It talks about how the earth changes through natural processes of erosion, of tectonics. And the old view of the earth was, of course, it was created at once, and it's like this, and maybe there's a flood, but otherwise nothing changes. And he shows by observation and evidence that none of this is true. The natural processes that change the earth. And that book was a big, big influence to Charles Darwin, who actually took that book on his voyages to the Galapagos, and read these constantly. In fact, The Origin of Species is dedicated to Lyell. So he is another big influence, because once you understand that there's processes that are creating change on Earth, well, then it's not such a big leap to say, well, maybe processes are also affecting species. So when people ask Darwin, what are you working on? He would ask, I'm working on the species problem. And the species problem is to understand, why are some species so different from others? Why are species sometimes very similar to each other? What creates the divergence? So in 1844, he wrote a letter to J.D. Hooker, which was essentially the most famous botanist in England at that time. And this is actually 15 years before the publication of The Origin of Species. And he writes, at last, gleams of light have come, and I'm almost convinced, quite contrary to the opinion I started with, that species are not, and it is like confessing a murder, immutable. So what's important about this passage in this letter is 2 things. First of all, he shows himself to be a true scientist. He didn't start out with the idea that he finally came up with. He said, on the contrary, it was the evidence that convinced me that I was wrong and I changed my mind. And second of all, he says it's confessing a murder. Why? Because he understands that this is going to get him into trouble. Because he understands that the consequences of this theory are going to go so contrary to church doctrine that he might lose his life over it. And like I said, this is 15 years before publication, so people sometimes say that was really the point in time where he had starting to understand things. But then he spent another 15 years writing about it, documenting about it, making sure that he really knew what he was talking about. And it was only because somebody at the same time had a similar idea and was about to go to publication that he finally published his book. So here's a picture of Charles Darwin. This was actually taken in 1854, and so it's 5 years before the publication. You usually see pictures of Darwin with a long beard and so on. Like that was not what he looks like when he was working on this. And here you see a picture of the first edition of the book. People have said that this is the most important book ever written in the history of this planet. By the way, if you are interested in obtaining a copy of the first edition, you can go around the corner to Sophia Rare Books. Apparently they have a copy that you can have for a cool $1 million. I once, many years ago, tried to actually buy a copy like this on eBay. I put it in the bid of $30,000, but I was chair of the faculty, so I had to go to a faculty meeting. And when I came back, it sold for $65,000. So it would still have been a pretty good investment, but So it's an interesting book. So what's in it? What's in it is his theory boiled down here on the slide with 3 points. Like, they're almost laughably obvious once you know about them. There's no math I'm going to show you. I'm going to just show you 3 principles. The principles are inheritance, variation, and selection. So what is inheritance? It means that some characteristics of an organism are going to be preserved from ancestor to offspring. Variation means that, in the process of generating an offspring, there can be some modification of these characteristics. And selection means that these characteristics have consequences. So in other words, some good variations make you have more offspring and therefore will be amplified, and some bad variation will make you either infertile or have fewer offspring, and therefore these characters will be suppressed over the generations. That's it. But to actually come to the realization that these 3 principles alone give you all of this biosphere that I showed you, well, that's not so simple. And in fact, that's why we're still working on these things. So once you realize that, then you understand that, well, that's a great theory. But it's a theory. Can we test it? Because that's normally the process that we have to go through. Theory, experiment. And the answer is absolutely you can do that, because you can do experimental evolution. And if you think, well, I've never heard about that, well, you should, because in fact, the first evolutionary experiment occurred during Darwin's lifetime. So here's the fellow that did this. It was the Reverend William Henry Dallinger. He was a microscopist who specialized in documenting certain microorganisms called amoeba. They are technically single-celled eukaryotes, so they're not bacteria. They're more complex than that. But he really specialized it, so he drew them very often, and like all kinds of detail. And at some point he realized, I could be using those to test Darwin's theory. He had read Darwin's book, right? So what he did is he wondered what would happen if I slowly changed the temperature in which these amoeba are swimming in and raise it. Would they be able to adapt to higher and higher and higher temperatures, which is something that Darwin would have predicted. So he constructed this contraption to do this. It's a really ingenious construction. What you see there is there are 3 vessels that carry the microbes. The eukaryotes are also called microbes, not just bacteria. And he put 3 different amoeba in each of the vessels. Now, they were not very different, but he could actually tell that they were different by looking at the microscopy, so that if one microbe would actually somehow infect the other vessel, then he could find out about it. He just wanted to have essentially a triplicate to get some statistics about the path of evolution, right? So you have these vessels there. Here's a burner down there, a thermostat up there. The nutrient media was circling through it. And what he observed was this. He started out at 60 Fahrenheit, and his plan was to go to 185 Fahrenheit, which is basically the temperature of these hot lakes, right? And so at first, he was able to change it and raise the temperature fairly regularly, like every week, right? Up until the point where he couldn't do it anymore. Essentially, every microbe would die. And then he would try to wiggle it up and down, up and down. Nothing would work until someday, like, oh, I can raise it again. And then he could raise it for weeks on end until he reached another one of these plateaus. And at the same time, he would look at them from under the microscope and realize, hey, they're changing shapes. Something is really going on. And then, it would go on again and again and again, and then another plateau where nothing would happen for a year. Right? You have to be very precise. This is a 7-year period. And then, in fact, at the last moment here, he still reached a plateau. But what happened is the following. So Dallinger wrote to Darwin in the middle of the experiment, and Darwin answered and said, I did not know that you were attending to the mutation of the lower organisms under changed conditions of life, and your results, I have no doubt, will be extremely curious and valuable. The fact which you mention about their being adapted to certain temperatures, but becoming gradually accustomed to much higher ones, is very remarkable. It explains the existence of algae in hot springs. So there was dialogue between the two, One had created the theory, the other one is setting it up an experiment, exactly like everything should have been. Except then, the incubator exploded after reaching 158 degrees before reaching the goal. He reported on this in 1887, and he writes at the end of the report, "Here, with such pain as I presume is natural, I have to close the story. The accident happened, destroying the use of the instrument and causing the whole to collapse." He never wrote another article and essentially stopped doing science because he was so invested in this. It was his life's work, and it had all gone up in flames. And to tell you the truth, a lot of people working in evolution today don't even know of his contributions, even though I think this was really a phenomenal attempt, even though it did not end exactly as you wanted. But 110 years later, there's another experiment that is actually still ongoing, and this is the Lenski experiment. He actually is doing experiments not with amoeba, but with bacteria. And the reason for that is they are much simpler. And if you want to understand the fundamental principles of something, you don't look at something that has all kinds of bells and whistles. You try to understand them by looking at the very simplest thing so that you can really get down to the fundamental principles. So he had not 3, but in fact 12 independent lines evolving in these Erlenmeyer flasks. And essentially is changing the nutrients every day, well, he and his students and postdocs, so that you can get about 7 generations of growth in a single day. And after 37 years, basically there are over 80,000 generations. And it's an absolutely fantastical resource because he can actually freeze all of the previous generations. And so you can actually go backwards in time and understand, how things have changed. It's an incredible resource that has validated Darwin's theory at every single turn. Not only that, in fact, it even has given rise to a new species of E. coli, which you can see over here. This flask is very, very cloudy. And the reason for that is that there is something in this medium that E. coli shouldn't be able to eat. No E. coli on Earth has ever been able to eat that. That's why it was in there. But this species evolved to metabolize it. And because there was so much energy in it, they grew up in such high numbers that they're all cloudy there. It's literally a new species of E. coli. And that evolved after about 35,000 generations. No other population, by the way, evolved that particular thing. So now that we know that not only is this a good theory, it's also validated by experiment, what then? Well, I want you to think about it. I showed you the 3 principles. Did I talk about nucleic acids and amino acids or any particular chemistry? And I did not. I talked about processes. So that means that, well, evolution shouldn't really be tied to any particular chemistry. So it really should be universal. Because the 3 principles, because they do not refer to chemistry, they should occur anywhere that the 3 principles can be at work. You can test, by the way, that if you take out one, the whole thing doesn't work anymore. Any single one take away from the 3, it doesn't work. So they're really not only necessary, but they're also sufficient. So what you can actually realize is that these principles can be formulated entirely in terms of the concept of information. That's where I'm starting to merge the 2 concepts, evolution and information, that we've been talking about. So you can ask, what is it that if it's evolving? People always say, like, organisms evolve. No, organisms don't evolve. It's, in fact, evolution is a process that acts on information. The information, of course, you know where it is. It sits in your chromosomes, right? There, you've seen them before. There's 23 of them. And if you want to see where the information is, you kind of have to go like, okay, so there's a cell nucleus. The chromosomes are inside of the nucleus, and they are a strand of DNA that's highly, highly wrapped up. But if you untangle, this, you'll see this double helix. And then if you try to interpret what's actually there, it's just letters. 4 different letters. 3.1 billion of them. That's where the information is. And that is what evolves. So I said the information evolves. But I mean, what do I mean by information? Well, for a long time, people just used the word information without really knowing exactly what it meant, until one man came along, the man that in fact created the theory of information, single-handedly, pretty much in a set of 2 papers published in 1948, and that's Claude Shannon. So that is the 20th century part that I'm talking about. He was interested in the process of communication from a sender, that's here the microphone, to a receiver, which is here the speaker. And he did this in a mathematical form, so he basically said, look, I'm not even going to talk about arbitrary letters, I'm going to talk about bits, zeros and ones, because that's the most fundamental thing that you can talk about. And I can ask, well, if there's noise on the channel, you know, crackle, you know, what happens is that some of these symbols get corrupted, and they might flip. So here a 0 was flipped into a 1. And he was asking the question, if I have high amounts of this noise, can I recover all of the information that was actually in the original encoding perfectly? And current wisdom at the time was no. At some point, the noise will just simply overwhelm it and you cannot reconstruct it. And then he showed mathematically that indeed you can do this error correction. In fact, up until levels of noise that are so large that the noise essentially completely erases all of the information and randomize it. If there is a tiny little bit of information left, so to speak, you can reconstruct all of it, which to some extent was a sensation. But it relied on a fundamental invention of some mathematical formula that allowed you to quantify the amount of information. And it was the first time somebody had did something like that, and it's the last time. There will only be one theory of information, just like there will ever be only one theory of evolution. And one was discovered by Darwin, and one was constructed by Claude Shannon. So, I'm not going to do any math here for you, because math is not necessary for science communication. I use it to help me think, because I'm just not smart enough to think with my eyes closed and just imagine things. I need math to sort of help me through it. But I don't need it to communicate things. Once I understand them, I can just tell you about them, right? Because you don't have to do the work that I did in order to understand it. So I can say it in a single sentence. Information is that which allows its holder to make predictions about some other system with accuracy better than chance. So information is that thing that allows you to make predictions. Making predictions is a powerful thing. If you could predict the stock market tomorrow, you'd be rich beyond belief, right? Generally speaking, in biology, if you're an organism, if you make better predictions, you survive better. If you can predict where the prey is going to be in 2 seconds, you can head them off. Prediction is survival. If you have 2 organisms fighting, one has more information than the other, the one with more information will win. So, in other words, information gives you fitness. And once you can actually understand the relationship between information and fitness, and we now have formula that can do that, but these are very modern formula from the 21st century, then you see the relationship between evolution and information, because fitness, which is basically the likelihood that a particular lineage will survive for a long time, that is essentially directly coupled to information. But not just genetical information. As we grow up, of course, we learn things. And information is stored in our brain. And we use that information to make better choices and better predictions. So information is a central concept that actually allows survival. It has innate information in our genes, and then there's information that we acquire in the brain. And all of them can be mathematically described. And their ability to increase your fitness can also be mathematically described, but I'm not going to do this for you, because this is all you need to know. So what we are really trying to understand here now is not the evolution of molecules, but the evolution of information. So let's take another look at these 3 principles, and now formulate them not in terms of characteristics, but in terms of information. And then you realize, oh, inheritance? Well, really, that's just a duplication of information. So the ancestor has some information, and then the offspring, is essentially having a copy of it. That's the inheritance concept, but written in terms of information. Variation is, of course, just the modification of that information. The thing that you just saw, that sometimes in the transmission of information, errors would accumulate. So that creates variation, but it's just modification of information. Selection is, in a sense, the meaning of the information. So in other words, if you have more information via the variation, then that information will be amplified. If you have actually less information, then in fact, that is detrimental. So everything now, the three principles of Darwin can, in a modern way, just formulate it in terms of information. And it can be done mathematically precise too. Now, maybe some of you might think like, well, Chris, if it's so simple and information evolves, why can't you just look at information inside of a computer, because our computer is full of information, and observe evolution inside of the computer? And the answer is, well, yeah, we've done this for a long time. We can create life inside of a computer by populating it with self-replicating programs. These programs make copies of themselves, the replication of information. They also have an error-prone copy process, which creates variation. And they live in a computer where this information is meaning. We started this 30 years ago in my lab at Caltech. It's called the Avida Digital Life System, and it actually now makes it possible to conduct extensive evolutionary experiment, not over 80,000 generations, but millions of generations. And we can do this in absurd replicates, not just, let's do it 12 times and do statistics on it. We can do it 1,000 times on compute clusters. So we can get astonishingly accurate predictions, or compare the predictions to existing theory, where nobody's going to hound us, about ethical questions, about all these computer programs that we are killing. So you see a computer program here. It's actually circular, just like the bacterial genomes. And there is an instruction pointer that sort of like reads this, and goes round and round and expresses the code in the sense that now the program does something. And they are, in this case, on a grid, and you see that they're colored in different colors, which are the different species. And now they're all essentially competing for energy and for resources. And basically what this looks like is you see, and this is very, very accelerated, you see new species emerge, old ones going extinct, competition between them. But in the end, the fact that we can create life inside of the computer, it means two things. It means we have understood life on a much more fundamental level, because now we can literally create it from these principles. But we can also now use it as an experimental system that's a lot cheaper and far more accurate to actually discover new principles about evolving systems. And we've done this many times. We've actually observed effects in this computer world. And we're like, well, that's interesting. I've never seen this before. But now that I've seen it, this better exists in the real world. And then we looked for that effect in the real world, and there it was. In other words, if that happens, you've created a system that captures the essence of what life is. Now, you can also ask me, so, well, fine, so we've understood evolution, but can we learn something that will actually help us as a human species? Can an information theoretic view of life and evolution help us fight disease, for example? And the answer is, I think so. And I'll just give you a short example from cancer. It turns out that many forms of cancer are due to, in fact, an erroneous flow of information. What do I mean by that? Well, an oncologist once told me, every morning a cell wakes up and contemplates suicide, only be talked out of it by its closest friends. So what do you mean by that? He meant that in a multicellular tissue, there is a strict regulatory code where no cell is allowed to have any individuality whatsoever. In fact, whether or not you live or die will be discussed by your peers every day. And only if they all agree, you're fine, don't kill yourself. They go, okay, fine, I'll go to the next day. But it's not just that. Cell suicide is apoptosis, and this talked out of by friends, that's just cell signaling, right? So it's like adjacent cells that says like, you look like you're doing okay, fine. But if you do something weird, you know what? We're going to withhold that, and so basically you're done. And that's a very important thing, because in cancer, that's when a cell is trying to regain some individuality and goes like, you know what? I'm going to do my own thing. So cells not just can't decide whether they get to live or not, they can't decide by themselves whether they can replicate or grow. They need growth factors to do so, which is also given by adjacent cells, by committee, so to speak. So I'll give you an example of a pathway, which is one of the most important pathways in cancer. And I know you're not cancer biologists, so I'll just explain a few things. Here's a cell membrane. And this is super abstract, right? But here are receptors. They're called growth factor receptor. So you, from the name, realize that, well, you know, there can be growth factors that can be bound here. And here's a protein called RAS. The pathway is named after that because it's the central signaling switch. And you also see a molecule attached to it, which is called guanosine diphosphate. You've maybe heard of ATP and things like that. That's a similar version, but just instead of a G, of an A of G. It's a signaling molecule, but you need to see that there's a D here, which means diphosphate. When RAS is bound to GDP, then essentially it goes like nothing going on, nothing going on. And doesn't send a signal downstream to grow. So that's a normal RAS when there is, in fact, no growth factor. It's healthy. If there is a growth factor, so the weird thing that happens then, in fact, you just saw maybe that these two receptors actually moved close to each other. And when that happens, a process happens here, and it changes the GDP to a GTP. So it's called a phosphorylation. It adds a phosphor group and makes GTP out of GDP. And then all hell breaks loose. The signal is being sent, and it goes like, go ahead, replicate. But now I'm going to show you the disease, the cancerous pathway. In the cancerous pathway, even when there is no growth factor molecule, this GTP hangs out there, and basically RAS is on, and it starts growing and growing and growing. This is, by the way, the pathway that is responsible for over 30% of all cancers. So we better understand this pathway. If we can fully cure this, the world will change. But what's going on here? So what's going on here is that, in principle, this GTP should be turned back to GDP by the action of RAS itself. So in other words, this is a switch, but if nothing happens, it will go like, okay, I'm off. And if it's off, there's no cell signaling. The diseased form basically goes like, I ain't listening, I'm going to do what I want. It's stuck in the on position, and it's growing and growing even though there's nothing, no permissions given. Now you might say, but shouldn't these things be killed by the apoptosis? And the answer is, in many cases, yes. You need a few other mutations in the apoptosis pathway. So cancer really requires multiple causes. But that's the central part of it. So how do we understand the flow of information here? Because clearly it's a communication thing that's happening. So it would be great if we had the following. So we have here the origin of the sensor. And over here we have the signal given. And here we have a bunch of different proteins, right? And it was great if we could just follow the flow of information like this. Right? Then we could go, all right, if it goes awry, then I can fix it, right? Except that's not how it looks like in the vast majority of cases. In fact, it kind of looks like this. So you give the signal, and then you see this, and then magically this thing turns on. And you go like, well, what went on here? And nobody really can answer this in the people who are studying this. But I give you a hint here. Take a look at this particular molecule as we look through this again. So let me go backwards, and let's look at it again. But now focus on this down there. So you see this just blinking, right? But it looks crazy. But now, imagine I'm only looking at the slides where this thing is in blue. What you then get is this. So now you can actually see the flow of information. But if you didn't know that only looking at the cases where this one is on, you could never find it. And in general, you don't know which one to look at. This is a case of encryption, a classic case of a signal that is encrypted, so everything looks random. But if you have the key, then you can actually find the information. And in this particular case that I constructed for you, the key was this molecule. If you knew how to just look for the on states, you see the information. And the reason why people have not understood this for the longest time and always said, we can't find the flow of information, it looks all random, is because they never realized that biology doesn't care if information is encrypted, so that we can't read it, and you have to find the key. So that means if we can find the key, the flow of information can be deduced, and then we can do something that people have not been doing before. What they've been doing before is they're trying to design a drug that attacks one of these circles. Like maybe if I just knock out one of those, then I'm restoring the flux of information. And you go like, that really shouldn't work like that because it's just a part in the information pathway. You're probably just destroying it. So what you want to do is once you find out what the flow of information is, then instead of knocking out targets, you try to restore or guide the information flow into a way where it has been before. Like for example here, you want to restore this particular path where in fact RAS is sending information to GTP and then essentially get rid of that. So in other words, it's a completely new paradigm of treating disease where instead of attacking targets, you try to understand and then reconstruct the flow of information. A 21st century application of information theory to biology. Now, you might think that, oh, Chris, don't people know about this already? Because here's an article from 2008 where it says life, logic, and information. But it turns out that these people didn't know at the time how to do the mathematics of information and of cryptography and things like that. And so it's really now that we have understood how to measure and quantify and find information that we can actually move into a new era. Now let me switch gears and talk a little bit more about life and evolution elsewhere in the universe. Life itself has an information theoretic definition. Namely, life is information that maintains itself. And you might go like, what do you mean maintains itself? Information doesn't get destroyed? Well, in our modern world, information is essentially permanent, but because we've made it so. Before we had books and computers, information was super, super fragile. It's unstable, and without a way to copy the information that essentially keeps it alive, it would disappear. Like you just put something, scratch it into sand, and it's just going to be gone within 15 minutes. So once we understand this, then instead of asking about the origin of life, how does life originate, you change the question, and you're saying, how does information originate? And it turns out that's a difficult question. Why? Because that needs two elements to do this. It needs to have information, and the means to replicate it. Here, in fact, you see a 15th century copyist just copying books. Why did they do that? Well, first to disseminate, but also to make sure that there's always a copy around. In fact, they understood that a copy process will maintain information. But we don't have monks, at the very early Earth, 4 to 4.5 billion years ago. So we can ask, what came first, the copy machine or the information? And the answer is, actually, we know it has to be the copy machine, because life's information is basically about how to make a copy machine. So the copy machine must come first. And then you go like, well, how could there be a copy machine on early Earth? And I'll show you one. This, what I'm going to show you now, is a possible scenario that I dreamed up. It is not what everybody believes, but I'm hoping once you kind of get the gist of it, you go like, well, that's at least plausible, right? It's called a plausible possible scenario. So imagine a clay surface. We had those 4 billion years ago. And here we're going to imagine 4 types of molecules, A, B, C, D, and they're like flying around. They're abundant. Now, in step 1 is you can get polymerization on the clay scaffold. That you can reproduce in the experiment. It happens. So essentially, this is what happens. And now that you have step 1, imagine that these molecules can form bonds. A binds to C, B binds to D. Biochemistry is full of molecules that can do stuff like that. In that case, you can get step 2, which is template-based polymerization, which is non-random. So that's what you just saw. You also saw that not all of the things are correctly bound, but the interesting thing is that the more correct bounds there are, the faster this process is. So like after this happened, like this thing can dissociate, and then if the new molecules come in, you made a copy. But these are not information. This is random stuff. There's no information in there whatsoever. This is just a copy machine. It copies whatever you put on it, right? But imagine the following happens. So you have this step, and then you have this step, and then let's imagine that in the third step, there's a piece that breaks off. And it just happens to sort of bind in this area. And remember, there was an error here. And remember, the fewer errors you make, the faster this process of template-based polymerization goes on. And now imagine that the action of this molecule is actually to make sure that it correctly binds there. We've also observed things like that in the lab. Now what happened here is that this sequence, with this thing breaking off, is replicating more faithfully, so it's replicating faster. So this sequence now actually gets an advantage over those that don't have this. That means that they now have a little bit of information on how to replicate this sequence. This fragment accelerates replication because it's more accurate, and therefore it represents a tiny bit of information about how to replicate. So what happened here is that the information about how to replicate has seeped from the hardware, this clay scaffold, into the software. And now you just have to imagine that many steps like this go on, and at some point, enough information is actually accumulated in these sequences that they can get rid of that clay and get rid of this copy machine, and they have their own copy machine. And so that means that once you think about the process of the origin of life in terms of information, you really can come up with plausible scenarios, whereas right now, if you just think of it in molecules, we don't even know what the chemistry was like 4 billion years ago. We can make very little progress on that fundamental question. But can we actually use information theory also to detect life on other planets? And I'm going to make this now very short because we are reaching our time limit, so I'm just going to give you the gist of it. The answer is yes, because what you can do is like once a molecule encodes information, it has to do it in an alphabet. And once there is information stored in an alphabet, that means that the distribution of the frequencies of the different molecules, like here, for example, I have 4 colors. This distribution reflects the constraint of the information. So in other words, if you see something that has very unnatural distribution, you're going like, oh, you know what? I bet you it's because there's information stored. We call this the monomer abundance distribution biosignature. For example, in English, this is the distribution of letters in English. This is the distribution for random, typewriter where there's no information in it. And the distribution is dramatically different. And so if there is an alphabet being used and information encoded in any set of molecules, we can probably detect that as long as we can detect the abundances of molecules on a particular planet. And what I'm going to skip over is basically how we showed that with molecules collected from a synthesis experiment in gray, which is like the Miller-Urey lightning in a bottle experiment to see if life can just sort of emerge. A meteorite, and then sediment was just, soil. And you see that the distributions are radically different. So we see, the only thing that really doesn't appear here a lot in sediment is beta-alanine, which is actually not an amino acid that is used in us. So there's thousands of amino acids, but these are the ones that are hyper-used, and as a consequence, we know that the distribution looks completely different. You can also do that with molecules that are not directly encoding the information, but instead, these are carboxylic acid, just are a consequence of that. And again, you can do the same thing, and you see that it's a perfect life detector. It works really well. So having seen that information theory cannot just only allow us to understand possibly the origin of life. It may even allow us to detect life on other planets, simply because we're now not looking for the origin of life, we're looking for the origin of information, and we're looking for detecting information, which is a much, much more general and universal thing. So let me leave you with a paragraph written by Lauren Isley, writing a book called Darwin's Century. It was written almost 100 years after the publication of Darwin's Origin of Species, where he makes the point that this work, this idea, is possibly the most important idea that ever emanated from this civilization. He writes, "It is my genuine belief that no greater act of the human intellect, no greater gesture of humility on the part of man has been or will be made in the long history of science. The marvel lies not in the fact that bones from the caves and river gravels were recognized with trepidation and doubt as beings from the half-world of the past. The miracle, considering the nature of the human ego, occurs in the circumstance that we were able to recognize them at all, or to see in these remote, half-fearsome creatures our long-forgotten fathers, who had cherished our seed through the ages of ice and loneliness before a single lighted city flickered out of the darkness of the planet's nighttime face." With that, I end, and thank you for your attention.